G'day, good morning. Thank you for listening. This is a MedCan show. I respect the law and I hope you do too. Today is an open topic, so nothing's really been organised. So if you're re-watching this back, actually, this information is for educational and scientific purposes. The information is fact-checked, and any papers or topics or pages like that I go through, the links will be put in the description below for you to peruse later on. And today is an open topic. I'll start at that bit now. So I'm hoping that these cool people in chat can put something in the description, in the chat box for a question or some sort of topic, and it can start the show. That's how I get the title of it. So if you're rewatching this back, you might want to fast forward a bit until you see some slides at least, or at least like the screen change, and that's an indication that the topic has been addressed or started. So I'll give it until 2 minutes. It's at 1 minute and a half now. And I'll start thinking something to say. It's a minute 40, 20 seconds to go. Mm, 10 seconds. I gotta say something. I'm thinking. Well, I can say g'day because 420 Fred's rocked up. So hello to 420 Fred. And actually, I know I'll go into cuttings because that was there's a question last week that was asked on that I didn't end up addressing. So if something else comes in the chat soon, good. Otherwise, I reckon I should start and elaborate on cuttings. It's like a tissue culture, because it's all it's wanting to do. So, all right, I'll just start on that then. So let me share the screen. I'll go into some plant science stuff. Actually, I'll go into plant development. How's that? So here we go. So plant science, no, we'll go into plant development course that I did. These are all the courses I've done related to plant science, soil science, and microbiology. No, plant development and biology. So a cutting. So most plants are totipotent in nature, meaning you can take cuts pretty easily from them. And there's different areas in the plant where you can take the cuts from to promote roots to come out and to have another cut and have another plant. Branching gene. This, there's a better slide than that. Characteristics of plant growth development. No, come on, I'm trying to find it. Oh look, there's circled, totipotency. Means does the plant have the potential to regenerate? So there's different sections you can take the cut at to get that to happen. And you're really looking at meristems. So around where the nodes are, that's where the roots are going to come out. So when you're taking a cut, so you could picture enough to zoom in, not really. So you've got the plant there, and see it's a node. So you'd cut just below the node on the angle. The reason why you cut 45 is just so it's not horizontal and it can allow water to create its little bubble, and then bacteria and fungi can germinate inside of it and create problems. So by putting it on the angle, it just allows that not to happen. And below the node, it's perfect because this in the node has cells in it that are undifferentiated yet, and they're meristematic, meaning that they can change into anything. So once it's cut, it'll promote the plant to start putting out roots, and if you can give it some auxin, which is the rooting hormone, it will promote that growth, especially when it starts. So that's about the easiest way to take a cut. What's all this say? Ovary, so meristem reprojection, tissue repair, auxiliary shoot growth, de novo organogenesis, lateral root formation, and meristem reconstruction. Oh, right. Just the different, they're not all meristems there. That is, if the plant's old enough in the internodes, it is. You've got ones in there that make the, as the plant grows wider, it has to have meristems in there to promote that growth. And down on the root apical meristem has got some right in there too. So that's where you take it. So if you, I'll stay on topic for cuts. What's this say? Plants are sessile, prone to physical damage. They have evolved developmental patterns that are continuous, responsive, and environmental cues and regenerative nature. These features are obtained by exchange of information between the walls, which can be local as well as long range. Cell fate in a developing region is more dependent on the extrinsic information as compared to intrinsic information. So it's more relying on the exterior information than interior, to cut a long story short. So you've got to create a good environment for it. So what's that? Approaches to study the plant development. No, this is a course, remember, so it does go into detail, and I'm just trying to cover a topic. Identifying desired developmental phenotypes. Oh, no. Identifying desired. No. Approaches to study a plant development. Getting away from the topic. Maybe that's a good idea. I should stop and see if someone's arrived. There we go. Sunshine has. G'day, Sunshine. Do you have any questions? Please put the questions or topics in the chat so I can see them and I can start and elaborate on them if you have any. That would be good, because I don't really have any. I have a cutting one from last week, which I started to, and I've pretty much covered it all, I think. I could elaborate it a bit more, but I don't know, it's not much to elaborate on in that sense. Maybe once you've taken the cutting, then what do you do? Sunshine Rainbows, he's into lab stuff. Yes. Nothing like tissue culture in a lab, is there? He's going, no. I just didn't fill all the words out. Lactic acid bacteria, which is more so what I think you're saying, hey. So for the cuts, yes, lactobacilli, lactic acid bacteria. I'll just, yeah. I'll finish this by, I'm already sharing, sorry, by seeing if there's anything else to taking a cut, because this is getting into the genomics. Okay, totally off track. And transfer genes, that's not the right one. To finish it, you want the environment of the cut to be put in an area where it likes. So it's got to be humid. What's this? So when you've got your cut that you've taken, oh, this is a bit better explained. The different areas to take it from. Oh yeah, and I can also go into the genetic mosaicism hypothesis, which is, I should probably pull it up then. I'll just pull it up because it's a good paper. And it has a similar diagram to this. This is just showing the areas of the plant, I suppose. Shoots on. What's the top say? Characteristics of plant growth and development. Oh, okay. Just characteristics. It's pretty basic. But it did lead to the, oh yeah, here's the bits that reproduce. That's pretty cool. Sorry, genetic mosaicism hypothesis, which is pretty important to this. That would be in plant science under genetics, I think. And it's a paper. Here we go, 2021. Put this in the description below. The accumulation of somatic mutations leads to genetic mosaicism. And really what it's saying is, depends where you take your cut on the plant as to how much epigenetics has been influenced in that cut. So if you take it from the top, there's a high, everywhere else there's a high chance except for in the middle, just below the middle. Cut a long story short without going through this big paper. So if you want epigenetics to have its effect, in other words, if you've had problems or if you've got a new variety to your area and you might want to adapt it to your heat or to your conditions, you'll definitely be taking a cut from the top, because that will allow those epigenetics to have manipulated the genes inside of the plant so that it will adapt to the environment better. But if you want ones that hasn't changed, because remember you're altering the phenotype in that sense by doing that, so you might get different expressions. So if you just want something similar to what the original was, you'd be taking it in the middle to lower, about there in the page where I'm circling. And if you'd like to go through this excellent paper, I'll put it in the description below, and you can later on. But it's yeah, that's the summary, I suppose, which is pretty cool. What else is down here? Just in case anything else rad. Yeah, so that's something I didn't think about until that first diagram, so that was really good. No. So yeah, anyway, I'll put in the description. Oh, it's a PDF, isn't it? I'll get this DOI. I'll put this DOI in because that's what I'll just click on it now. And hopefully it's, well, anyway, it'll still come up with a website. So I'll put this in. And then you can go and download it if you want. That's how I got it. He's cool. Professor Devaud from Canada. Very good into. And what's the other thing to talk about? So I'll leave that there. Some sharing just in case. Genetic Farms has chimed in. G'day, Genetic Memory Farms. Nice to see you, mate. Genetic Farms says, so I have to confess I have sold out. And will now retailing at a legal dispensary. Very good, mate. Very good. That's good. And the last thing of the question was of taking cuttings was the environment that you want to put it in. So now you've known where to take it on the plant and how to take it because of the meristem action and the environment that it wants to go in. So you can dip it in oxen, IAA or NAA or a good oxen to promote the stimulation of the roots, or you can just leave it. It's good to cover the wound though, because you have created a wound. So you can even put honey, aloe vera, all that sort of stuff on it. But as long as oxygen hasn't gotten in, so you're not going to take a cut and leave it on the bench, and then it's going to try and absorb light and go through its process. So instead of root pressure, it's going to suck up oxygen. That's bad. That's really bad. So that's probably one thing you have to watch out for. Should be sharing, talking about stuff. And then the environment. So the AERO. So there's really cool environments you can put them in. So you can go and mist your environment. You can put a bag over the top to keep it in its environment, you know, it's moisture high. Or you can do these types of things. So this is kept in its own moisture environment, and it also promotes, it's got a bubbler in the bottom, so it promotes high humidity for the roots. So there's a good example of it there in the middle, how it's got just a sprayer in a circle, which just sprays out to create that humid environment, which doesn't get up into the top. So that's what I promote. One of those and you don't go wrong. Or you can put it in your substrate. You can just take a cut, put it in your substrate. You can leave it in water. You can do a few different ways to do it. Depends what suits your environment and how hardy the plant is before you've taken a cut. I didn't talk about that. You've got to make sure it's in good health. If you take a poor health, you're taking something without roots and you're going to take a cut and expect it to try and push out roots. It's got little energy and it's probably already using its stored energy for fighting its immune system because of its problem. So you have to take a cut that's in good health. So you can foliar spray even before that with some say liquid kelp a few times before taking it off, say 10 days before and 5 days before, and whack it in your environment. You're pretty much right. And it'd be good to have, if there's any questions, to see if they want to elaborate any more. But that'll do, because Sunshine would like some elaboration on lactic acid bacteria. Well, what is this lactic acid bacteria? It's I'll show you, share a screen. So labs, what is this stuff? Well, it is a families of bacteria. Does it go right up to family? Let's have a look. It's a genre. It's a genre of bacteria. Okay. It's very low there. Okay, that's pretty interesting. So it's a genre of bacteria that's very what's the word for? I'm trying to think of it puts out a biofilm. It's not very symbiotic. It's good used for milk because it's a good preservative for there. It's a good food preservative because it puts out its own biofilm, which is antibiotics to stop other bacteria from colonizing. But it can be also worked very good in plants. These bacteria are usually found in decomposing plants and milk products. They produce lactic acid as the major metabolic end product of carbohydrate fermentation, giving them the common name of lactic acid bacteria, or labs for short. So the genre has many species within it, So there's lactobacillus, which is common. You find in your yogurts and milks. Streptococcus, that can be not good, but it's still, anyway, there's a few of them here. I'm not going to read them all out. That was the pediococcus. It's a round one. Okay, that's all that means. They're either rod-shaped bacilli or spherical cocci under the microscope. So you can look under the microscope and see which ones you got, but that's what you're going to expect to see. Metabolize are classified for two main pathways of fermentation. Heterofermentation. Well, hetero means different things. So that would have different stuff in it. And it must be a mono. Okay. So members of lactobacilli appear to be able to perform aerobic respiration as well, making them facultative anaerobes. Because remember, the obligate anaerobes means it must do it. So this type is a facultative. And unlike other mammals in the order, which are aerotolerant, using oxygen helps these bacteria to deal with stress. There you go, sunshine. So the more oxygen, the better they're going to enjoy their environment. Because really when you're feeding the plants, you're trying to feed the microbes. Because the microbes will do the work for you if you're into organics, which I promote organics, not mineral feeding or synthetic feeding, making the microbes work for you. And in this sense, yes, your labs want more oxygen. So the more oxygen you can get into them, the better. Okay. Phylogeny, we sort of already covered that. Uses, probiotics, okay. Foods, very good. What about in agriculture? Fertilizer. Researchers have studied the impact of lab on indole acetic acid or auxin production, phosphate solubilization, and nitrogen fixation on citrus. While most of the bacteria isolates were able to produce auxin and phosphate solubilization was limited to only one of the 8. So there's something cool, also sunshine, that they produce auxin, so that it's going to be very good for roots or for clones, cuttings, those types of things. Very good. Lactic acid, fermentation, no. Want to do the, no, where's the agriculture? Doesn't have any. All right, so it's not very common in agriculture, but it can be used. Bacteriophage, no. Management of bacteria, no. Bacteriophages are things that eat bacteria. So they're really good to control problematic bacteria. So that's why, yeah, they use these as a broad number of food products, commonly chemicals and biotechnology products, are manufactured industrially by large-scale bacterial fermentation. Because this involves cultivation in enormous quantities of bacteria each day, a serious threat is that a risk of contamination by bacteriophages, which can rapidly bring fermentations to a halt and cause environmental setbacks. So yeah, it's got to be clean. We should already know that dealing with bacteria. Labs are used in food. I don't know, I'm not talking about food. Okay, well, that's about it for that one. I've got some slides where I can see if there's any questions. I've got some slides I can elaborate a bit more on. Robert Connell Clark suggests doing an iodine test on your cuts. Oh, the darker it stains, the more carbohydrates it contains. He said it is the amount of carbs present in the root which let it root. Yeah, well, because the carbohydrates of the energy are stored as starch, and that converts in the glucose cycle back to energy. Cutting not on. Yep. Yeah, very good. But you'd have to see Now what's the starch stain? I've got it. Instead of remembering getting it wrong, I'll put up here stain, microscope stain for starch. Good. It's backs up what you said. Where's the share button? I just wanted to verify it and see what the search engine said as well. And it's good. Sharing good. Yes. So you want to source Lugol's iodine, which is potassium iodide and iodine in water, is for Lugol's iodine. And then once you will drop it onto the tissue, it'll turn the surface of the granules dark blue. And then amylose binds to most of the iodine at about 20x more than amyloipectin. There you go. I thought it was going to be informative and help. That last bit didn't really, but that's what you want to see. So after you do drop the stain in, you'll see all the little round dots, and that's the areas that tells you where the starch is. Very good. How about images? We got anything cooler? It's always good to push that because you get different perspectives from different studies that different people have done. But look at that one. That's a pretty good microscope though. That's not your normal light microscope. Desktop, that might be. Looking at 20x. Oh, there's, yeah, you can add a few different stains to certain images and get the colors. That's pretty cool. So I put a couple. Anyway, back to the topic. Good information, thanks, Genetic Memory Farms. Labs, that's right. And I'm going to look in my plant science stuff, probably microbe section. Let's go back to microbes for lactic acid bacteria. Might even have a section on it. I do? Look at that. Oh, and I've got the one thing in it. Lactic cultures. So lactic cultures is a biopreservation technique with the use of lactic acid bacteria. This is used in post-harvest treatments to prevent the development of microbes that are responsible in shortening the shelf life and freshness of food products. This is an example. So lactic acid bacteria has the ability to produce antimicrobial compounds with strong competitive action against destructive microbes. So benefits generally recognized as safe, environmentally friendly, and decrease the rate of deterioration in fruit and veg post-harvest. They prevent the growth and activity of several post-harvest pathogen fungi in fruit and veg, extends shelf life of agricultural products, and reduction in microbial contamination. And they're compatible with most biopesticides, most bio-fertilizers, and plant growth hormones, but do not use with chemical fertilizer or chemical pesticides. So yeah, that's why I don't use them, because I've just been amazed that people can do so well out of it. But remember too, a plant has 96% of its nutritional requirements comes from the atmosphere. So it's only 4% that has to be as given it to it or be made available. So it's not a great deal to get an outcome of a crop. But I'm sure I got some more stuff in there. I should see if there's any because I'll have to search a bit now. Which other classes I remember I've done it in. Is there any under L down here? Yes, no. Doesn't look like it's alphabetical. Okay, what was it saying? Lactic acid bacteria, got to stay focused. The role of bacterial biofilms. Oh, that's for antimicrobial resistance. That doesn't really help in this instance. In my courses, it would have been, I know where I'll have to look through. I should stop, see if there's any questions on that. And then I've done a food microbiology and food safety course, and that was brilliant just for this, just to learn about microbes and how they function. Lactic acid bacteria, size, irradiating, antimicrobial food additives. They'll talk about it in there. But I want to get more. No, didn't really help either. I'm struggling a bit here. It's in here though. So I'll look at, oh, that's right. I'll stop first and see if there's any questions. Cutting stains. Genetic Memory Farm says it is even observable with the naked eye. Oh, related, talking to that stain test that you can do. Awesome. The starch stain test. Yeah. If your plant's in good health, you won't need to do that. But this is a good way. That's a good way to confirm it. Just to know, are you guessing? Is the plant bluffing you? Is there a hidden health? Is it going through where it's showing you good signs, but it's actually not in good sign, like hidden hunger, which is a thing in plants? Okay, back to it. I'm going to look through my course on food preservation, food safety. Fully shared. No. This one. So I have to skim through a bit. It was a good course too, by the way. Because look what it goes through. Microbial groups associated with food. It just touches on all that sort of stuff that you really I want to know about. There we go. Look at that. Second line. Lactic acid. So beneficial roles of microbes. Various strains of bacteria and fungi are used in fermenting dairy products like lactic acid bacteria, which are produced for coagulating of the milk. Lactobacillus and bifidum bacterium are used in probiotics. Survival. Air with low moisture, dust content, and high temperature characteristically are having a low microbial load. So by learning what microbes need and use and function and how they get their metabolism activated, it's very, very important, because for me, that's all I'm trying to replicate when I'm feeding my plants, because I'm looking at the plant's health from the leaves and their morphogenic characteristics, and then learning from that, all right, it's doing this. Oh, it's got a sickness. Oh, it's got a toxicity or a deficiency. And then I can rectify that by the microbes. So then I'll think, okay, these microbes, okay, it's got claws feet, so there's a nitrogen toxicity. Okay, I'll stop the nitrogen-fixing microbes, as an example. So by knowing the environment, what's it go in, it's very, very important. And this course showed that to me. I'm not going to go through it in that detail, but I'm just focusing on lactic acid bacteria. So I'm going to skim through this and just try and pull out the lactic acid bacteria highlights. Is this a change in pH of the plant tissue by post-harvest molds like botrytis or penicillin facilitates the growth of disease-causing bacteria? Yes, that's post-harvest operations. I'm not going into that. And that was a good course too, post-harvest agricultural operations. Yeah, did you know, like when plants, when you go to harvest, you cut them down, they're still alive. They mightn't have root pressure, but they've got cell pressure. Isn't that interesting? That's in post-harvest operations. All right, there's probably a couple hundred slides here. Cereals. No, I'm going to try and get to moisture content, which is everything. Water activity level. We are none. I'll stay on topic. I'm going to have to get, I haven't seen these for a while. This has got more stuff on the moisture levels. This is rad. Way more active water activity stuff. So it's above 60%. See, look at me getting excited. I should stay on topic. This says the actual ones. The other charts I've got go from 60 to 65, 65, 70. So aspergillus is definitely at 64. Rad. 61. Yeah, xerophilic. Back to it, lactic acid bacteria. That was the name. The different classifications that they can come under microbes. We're talking about facultative and obligatory before. Aerobes, facultative, and anaerobes. This isn't a microbe lesson. This is on lactic acid bacteria. Approximate pH values. Okay. Minimum. This is excellent. Look at this chart. The minimum, optimum, maximum pH growth range of some potential foodborne organisms. So in other words, it's telling you these microbes, if you use these, this is the minimum, optimum, and maximum pH range. Right. So max is all alkaline. Optimum, pretty much neutral except for yeast, is a lot lower. And minimums, look at that, how the fungi are low as. The bacteria, the minimum pH they operate is 4, and you'd probably nearly call it 5. So you can say substrates nearly below 5 or 4.5 pH don't really have bacteria. Isn't that interesting? Or Bacillus cereus, that's a bad one. That's anyway. Stay on topic. Lactic acid bacteria. I haven't been through these for ages. These are rad. Every 10 degrees Celsius rise doubles the catalytic rate of enzymes, and every 10 degrees decreases reduces it by half. Isn't temperature such an important thing? Microbial growth is accomplished through enzymatic reactions, which is dependent on temperature. Temperature growth of microbial curve is, isn't that cool? Optimum, maximum. And remember, there's the different levels. Mesophilic and thermophilic is the range that we're in, and also mesophilic, because thermophilic is above 65. That's the range where you want your compost to be in to break down all these problems. Great. And there's a mesophilic and thermophilic. If you really want it, the 30 to 40. Oh, it's 55. Sorry, I was wrong. Optimum between 55 65. And the psychrophiles are the cold ones. And look at what's in the psychrophiles. Lactobacillus. So these organisms will grow well at or below 7 Celsius, which 7 Celsius is about 40 F, and have their optimum range between 20 Celsius and 30 Celsius. So that's between 65 Fahrenheit and 81 Fahrenheit. So that's good for you to know also. Your lactose. So that's pretty cool, because the mesophiles, they like the range up pretty high. Yeah, optimum looking at 30 to 40, up around 35, 40. And as you know, having a substrate at 35 or 40 is not beneficial for the plant because of its oxygen uptake. That's all for that one. That helps with labs again. Oh, yeah. Is my screen on high? Yep. Temperature dependent. No. Antimicrobial effect on gases and microbes. Alrighto. Conventional. I'm just trying to skim through and get the ones with the labs and the points for the question, because the question wanted elaboration on labs. Quality of food samples to be collected by analysis. Okay. Media enumeration, that's counting. So this is pretty cool. So if you want to count your microbes that you have, you need a Nebular chamber or a hemocytometer, which is what I have. Hemocytometer, hemo is blood, cytometer, don't know, but that's how I remember it, and it's for counting blood. So you can use it also for counting microbes, because all it is, it's got a miniature under the microscope scale that you can read. So it's like a grid pattern there, and you put them into blocks and count them and do your multiplication out to know how much your count has. It's very cool, but that's not what we're into. Standard plate count. Righto, there's different ways to count them. Measurement of metabolic activity. So how do you know if your microbes are producing and doing the things that you want? You can do these tests. Actually, involves the utilisation of methylene blue. It's a beautiful stain to establish the policy. Measurement, no, not getting into that. And how to measure cellular constitutes measurements. So you want to know what's in the cells. Methods of food examination. I'll skip past a few because we're not talking about counting them. Immunological methods. So the reaction reveals the quantitative and qualitative conformation of antibodies and antigens within the cell. No, far out. You click it. No. Standard plate. Ethyl blue. No. A lot of tests here to test your microbes. So if you want to go back, you can just pause each one I've got here, each slide, and you can just read up yourself. Routine microbiology for other food and products. Standard plate count, coliform and yeast. Where's the E. coli test that you must do? Most probable number, water examination, it reckons. So it says 3 sets of 3 and 5 oh, that's for the, all right. It's not. Specific microbial methods for counting pathogens. Okay. These are more different ways you can count them. Stains. Very good. It'd be good if there was a lacto one specifically for it. I'll stop on that slide and elaborate on that one. Principles involved in preserving food. Yeah. Drying and ferment. No, come on. Refrigeration and freezing. Food preservatives will probably come up in here. Yes, many ways you can do it. Categories of preservatives by their type of action. Well, that's pretty cool. Pretty cool. Irradiation. Yeah, that just kills most of them. If it's at the right dose, the right rad. Yeah. Okay. I think it's been into it because this looks like they're getting out of the. This is different methods. Calculations. Okay. I'm trying here. Just going to try and go through them all because these are pretty cool slides. They're right on topic. Irradiation levels and examples of what happens at the different levels in food. It's nearly a topic, isn't it, on itself? Our LPs have to have large devices to do that. It's not acceptable. Advantage of food radiation, disadvantages. Yeah. Food or product loses its nutritional values because of the destruction of the vitamins and nutrients contained in foods and other secondary metabolites, and a few protein foods and flavor sensitive to irradiation may develop off flavors. See that? So by irradiating it, changes the end product. But who cares? It makes it safe for the human. It's terrible. I'll just go through slowly on that one. So if you want to pause it and look back later, read how it actually is bad. Oh, here we go. Lactic acid bacteria and its antagonistic behavior. So labs are a group of bacteria exploited since long for the production of fermented food due to the ability to produce desirable changes in taste, flavor, texture, along with inhibition of spoilage and pathogens. So they have a great, generally recognized as safe status, the GRAS. They are, I'm not going to read all that. They're resistant to bacteriophages, very good. And they attractive physiological properties and technological applications such as proteolytic activity, lactose and citrate fermentation, polysaccharide production, resistant to freezing and lyophilization, adhesion and colonization capacities to the digestive mucosa. And they got great antimicrobial production. Labs include genres like Lactobacillus aerococcus, Carnobacterium, Enterococcus, and many more. They are good biopreservatives as they extend food shelf life and inhibit the growth of pathogenic and food-spoiling microbes like bacteria, fungus, and yeasts. See that? They inhibit the growth of pathogenic ones, but they can also not work with good microbes, so don't allow them to multiply because the lactose put out a great biofilm to protect themselves, which has a lot of antibiotics in it. Lactic bacteria and its here we go. So there's the lactic acid bacteria cell, and it's got all around it. It just puts out its double layer through its gram negative, gram positive cells that it has. It's double layer cell walls, and nothing's going to get in because it's so strong with its antimicrobials and its biofilms. Propionic acid bacteria convert lactate to propionic acid and to CO2. So you might be working with these ones too, Sunshine, some PABs as well. So they're similar in nature as they can still do a similar thing. I won't really read into it though. Microgar, it's a brand, approved, inhibitory activity of propionic acid bacteria and its salts against mold and bacillus subspecies. They are accepted as preservatives for industrial use in bread manufacturing. All right, looks like we're off the topic now. Organic acids are organic labs through an oxidoreductive process, which is fermentation, synthesize organic acids as main end products. Some species like Lactobacillus, Lactococcus, and other labs have been used as a biopreservative in fermented food for long years. Isn't it? Anyway, the organic acids particularly secure the food by reducing pH of the surrounding environment, generating a selective barrier against non-acidophiles. the organic acids also proposes antimicrobial effects on pathogens like E. coli, Streptococcus, and Salmonella, and Listeria species, in especially meat and dairy products, also spoil with bacteria. So yes, that's why they're used. That's last one is the reason why they're used in foods. For that reason, they work rad against bad things. Lactic acid kills the unwanted microbes by disrupting the cytoplasmic membrane and by interfering membrane potential and the intracellular pH. So by good luck colonizing it with other microbes, acetic acid shows increased antifungal effect in the presence of propionic acid. Other organic acids have been found effective as antifungal biopreservatives. Oh yeah, that's just an interesting fact, not related to the topic. Okay, looks like we're off topic again. That was a good rundown on labs though, and PABs. Seeing foodstuffs. All right, we're into the gases now. Modified atmospheric. Biochemical fermentation. It's this table of ferments. Oh, it's got lactic acid bacteria based on kind of microbes. All right, doesn't help. All right, there's a shape. There's some lactobacilli. What have we got? Some here. I'll keep the photos up there because they look pretty funky. Some microbes involved in lactic acid bacteria plays a crucial role in desirable food fermentation, along with the lab group Lactobacillus and Lactococcus species, and most commonly used these species. Other important organisms besides labs involved in fruit fermentation are acetic acid or acetobacters. Wow, Gluconacetobacter diazotrophilis. Popular ferment has the names of the different types, the genre and the species of the lactose. There, if that helps you, if you want to really get into the different types that you have or different types that are available, because they'll do different things. Like there's another one, Lactobacillus plantarum. That's in general. Fermented food. Looks like you should be doing this course, Sunshine. Health benefits, probiotics. No, benefits. Beneficial health effects stated for probiotics. And look at them all. They're all the labs. So there's a paper you might want to put into your search engine and look at that, written in 2014. Successful probiotic organisms. Look at them all. So good probiotics. Value added food products. Okay. Here's the major subgroups in lactic acid bacteria. Group 1, Group 2, Group 3. Group 1, obligate homofermentative. They grow at 45 degrees Celsius, which is blimmin' hot. That's up over 100 F. But they're unable to grow it at 15 F or 40. Actually, it's 45. No, 10 is 50 F. Group 2, they grow at 15 Celsius, but show variable growth at 45. And Group 3 produce lactic acid as the major product formed by glucose fermentation with minor amounts of CO2 and ethanol. Okay. So then that must be group 3s, not temperature dependent. Then you want to break it down further. I'm not going to go into this, but you can see if you want to really get into different types of lacto acid bacteria that you have, you can do that previous chart. Some genre of labs, cell morphology, what they look like. So if you're looking under a microscope, you'd be looking for round ones. Some are chain and round. Some are rods like Bacillus and Pseudomonas. And that's what they do. Desirable characteristics of a starter culture. Wow. Shame we're not talking about that. It's got labs down here, look. The organism is effective both as a single strain as well as a member of a mixed strain starter along with other labs. Lactic acid bacteria production of lactic acid. So do you want to produce lactic acid? Is that what you're trying to do with your labs? I don't think so. I think they want it to produce the oxygen that it gets out of it and function somehow and make it help the plants. So I'm not going to read all that. What? Lactose is converted to lactate, which inhibits growth of undesirable flora. Okay. Flora and fauna. Lactic acid bacteria are the key organisms for food production. Very good. I'm trying to stay on point. There's little labs in the middle. Possible benefits of lactic acid bacteria. They inhibit pathogens. Oh, I can't even read that. Don't know. Stimulation of immune system. Anti-cancer activity. Stimulation of immune system. That's what I think that one is. Nutritional improvements in general. Okay. Bacteriocin. Lactobiotics. Heat stable. Heat stable. Heat liable. All right, that's just some different classifications. Not going to go into them. Primary and secondary microbes. This is more lab stuff. This is just if you want to manipulate your labs to do different things for you and different food products. That's what the genre you use. Okay. Vegetable based. Seems wow, there's a lot of them. Bacteriocins of lactic acid bacteria. Very function benefits of lab fermented food products and the health benefits. The health benefits study indicate improvement, promotion of the immune system due to the ingestion of the labs. Isn't that interesting? So if you're into extraction, possibly you might want to consider using labs to end up in your end result, which will promote the immune system. That's very interesting. This is attributed to their ability to activate macrophages and lymphocytes in an improvement in the levels of the immunoglobulin A and the production of gamma interferons. Few reports show that the ingestion of labs present in fermented milks exerts. They result in lowering serum cholesterol. Okay. What? This is dangerous. You don't want to. That causes poisoning, food poisoning. Bacillus cereus. Pumilis is rad. That's actually good for salicylic acid. Stay back on point. Labs. Functional benefit of fermented bacteria using labs. So the bacteria colonize human guts, resist low pHs, prevent pathogen growth with their antimicrobial activity, safe to consume and show no antibiotic resistance, suitable for commercialization. So see that? Show no antibiotic. So anti means against, biotic is living. So they show no resistance to other things. That's the end of that one. I should see if there's any questions. So I've still got one more to go through. One more. G'day, Raptor. There you go. I started on cuttings and elaborated on the cuttings. You might want to rewatch the first, I think it went for 20 minutes. And then I got into the labs. G'day. Raptor says, when I applied my labs, it got rid of my algae bloom on the side of my pot. There you go. Genetic Memory Farm says Lactobacillus plantarum seems to be the most common in brassica and nightshades when we get them in some salty waters. Oh, sauerkraut. Yeah, Cedarbacter for the win. Actually, I've got a Glucunacedarbacter diazotrophilis that's it's nitrogen fixing bacteria. Reminding me of to make ginger pop. Very good. Looks like Sunshine is still not here. That's a shame. Because all right, I'm not going to elaborate anymore then. Well, I suppose I should. Let's see. Just because he's not here. He asked the question. Doesn't matter. This is a very informative talk on labs. Like extremely. A lot of those slides. There was heaps of slides that I could pull out and put into my lab section now. So I'll go through and see if there's any more stuff in here quickly for labs. Here's cocoa bean. Oh, it's just different. Yeah, okay. Here's some Cedarbacters. It's a yeast. Oh, no, it's not. It's a bacteria, sorry. So I'll send these. Main yeast for Saccharomyces. Non-alcoholic, vinegar, yeasts. There's another lab, meat production, sourdough. It's all through it. But I really want to get on it's more around what they do. Many natural fermentations are carried out by collective activities of yeast and lactic acid bacteria. I'm just trying to get the point across on if you're going to use them, these are the parameters and what they are and how to get the best out of them. The contribution of labs in the fermentation of plant products. Bloody excellent. This is the one I should have started on. So you got your three types again: homeofermentar, facultative, homo, and obligate, hetero. So the contribution of the lab in the fermentation of plant products. Okay, that's the fermentating of them. So, for instance, sauerkraut, like GMF said before, that's what you'd be trying to source those different ones to do different things. This is a natural source, spoilage of food, food spoilage, probiotics, and biotherapeutics. Wow, it's a big word. What's this? Yeast, mold, labs. I think I did read that out before. Microbial constituents, mold, lactic acid fermentation, non-lactic and lactic. Okay, so you can get non-lactic fermentation. Oh, they're not any lactose though. So if you want to use if you use lactose, expect to get lactate. Dairy mold, lactic, acidophilus. Kefir. Types of starter cultures. Starter culture used in dairy and food. Wow, types of starters. Mesophilic starter culture, when optimum growth temperature is 30, or you use a thermophilic if the optimum growth temperature is 43. That's pretty cool. That's really, really cool. And what's this? Depending on the physical state, starter culture can be liquid, solid, or powder. I know, but I like that. Thermophilic 43 gives a number. Criteria for selection of starters. I know. Starter culture for preparation. Look at all the lactose. This is like a whole course on it. Starting concentrates. Yeah, what concentrate do you want? No, don't. No, where is it? Had the CFU there. It's not blooming it. There it is. So you want the DVI, direct VAT inoculation, concentrated cell preparations containing 10 to the power of 11 or 10 to the power of 13 CFUs per gram. Wow, that's high. So 10 to the power of 11 would be 10 billion? Because I use 10 to the power of 10. So in other words, you've got to have it up high for a starter. I knew that. Most concentrations of CFU, that's the colony forming units per gram. So if you haven't got many, the chances of it colonizing is lower. But they suggest here you should be using 10 to the power of 11 or 10 to the power of 13, which is heaps. Because if you look on the back of a lot of microbial containers that you'll get it in, for instance, Great White, they'll have the CFUs and they'll be in the million. They won't even be close to billion. And this is asking for 10 billion. So, for instance, if you'd like to use that product to get the effectiveness as this is asking, you'd have to use not grams, you'd have to nearly use kilos. So it's very... I'm starting a bio-fertilizer business and I'm focusing on 10 to the power of 10. But back to lactic acid bacteria. Backslapping. Artisanal cultures. Oh, GMF's eyes go high. Steps. Yes. I'm not going to go through all this because I'm really just focusing on lactobacilli, labs. An outline of various stages in manufacturing. No. Okay, I think I can at least stop this. I think we've covered just about every section of it. Now we're getting into pasteurization. Sterilization. List of different microbial groups commonly used in the production of different fermented milk. Bifidus types of fermentation milk. Wow, look how far back it goes. 6000 BC. Fermentation. They've been using these labs since 6000 BC. Classifications. All right, I want to stop it there. Let's say labs. That'll do. Oh, minute 9 too. Good. An hour 9, sorry. Reminds me, right, it says proprietary microbes. Yes, specialized microbes to do things that you want them to do is the go. I can talk about that in another show because it's just amazing getting microbes to work for you. And if you let them colonize everywhere, that's bio-fertilizer, biopesticides ongoing for free forever, as long as you look after your microbes. And that's pretty much on what today was about, looking after your labs. So put your chats and stuff in the comments in the description box now, because I think that's about it for today. We've covered labs really well. I'm going to change the topic of today to cuts, taking cuts, areas of the plant to do it on, and different criteria like that. I'm also going to retitle it Labs, Lactic Acid Bacteria. So I think there was some good stuff in there, and I've got those few papers to put in. I'll put in the description box below. And it was good. I really appreciate all the comments in chat, which makes the show. I hope you can bring all your questions next week and we can do the same thing again. I'll be doing this for quite some time. So save your questions and I can elaborate with some slides, I hope. And then all these new courses I'll be doing soon too. Next semester might be more agricultural engineering type courses, which would be fascinating. What does I have to say? A good example is how we process insulin since the 1970s. A very good example. Yes. Thanks, biotechnology. Exactly right. Biotechnology is brilliant. How to make living things work for you. How to multiply them, and it's so excellent. That plant science course was fascinating about biotechnology. Plant cell. Here, I'll just show you. Look at the biotechnology courses that I've done before we sign off, because that's one topic that really gets me going too. Study biotechnology, biomimicry. That was pretty cool. Environmental biotechnology, experimental biotechnology. There's got to be some plant biotechnology, plant bioprocessing. No, post-harvest, no biotechnology. But yeah, there's a few anyway. So it's just fascinating on how you can manipulate and make biology work for you. Oh, stop sharing first. So yes, I agree. Thank you, biotechnology, for allowing us to manipulate biology and make it work for us. Excellent finale. Thank you, everybody, again for listening, and thank you for you who contribute as well. I do appreciate that. And for the rad people in chat, can't forget them. And I hope to see everybody next week. Happy breeding, happy growing, and good health to you all. Bye-bye.